I'm basically going to give an overview of some work that's been done with collaborators, in particular two PhD students, Max Lau, who's now at Emory, David Thong, who's at Heriot-Watt, other collaborators are Colin Warby, formerly Princeton, and my colleague George Triptaris, along with Glenn Marion from BIOS and Brian Grenfell, and this has been done with funding from EPSRC and also from Biomathematics and Statistics Scotland. So a very quick overview of what I'm going to talk about. I'm going to be talking about spatio-temporal stochastic models for epidemics, and mainly I've worked in applications involving plant diseases. A large part of the talk is going to focus on a particular problem of how we try to incorporate pathogen genetics in with data on the epidemic itself. So it's really a kind of data fusion problem that I'll be looking at for part of the talk. But an important part of the talk for me, and something that I've been very interested in for a while, is the whole problem of trying to actually assess the quality of fit of epidemic models. So I'm going to look at a couple of ways of trying to actually try and test out models that fit within the Bayesian inferences that I'm going to be doing. So looking at latent residuals and also the idea of latent likelihood ratio tests for kernel selection. And so I'll try to illustrate the methods with some simulations and also some application to real data. So this model that I'm talking about has effectively been covered by other speakers in the conference, so I probably don't need to say too much about it, but this is a continuous time SEIR spatial-temporal model. And in this model, it's going to be individual based and hosts will be located at discrete points. To describe the model, well, the main process that we'll be interested in, the process whereby susceptibles become exposed. So in this model, essentially the instantaneous rate at which some susceptible located at J is acquiring the disease is going to be composed of essentially a primary infection process, also called the Sparks term sometimes, so that will be alpha. Then I have a parameter beta, which is an overall secondary infection rate. And then importantly, what we're going to do is we're going to kind of have the infectious challenge from an infected individual I to my susceptible J. That challenge is actually going to depend on the distance between I and J. And as I've written it here, this would be an isotropic kernel function, but this can be done for anisotropic kernel functions. But the K is simply a function of the distance. And then to complete the model, I need to specify the sojourn times in the E and the I classes. So I would assign some distributions to that, and typically we use gamma and variable distributions. So that would be my parameter vector that I would have here with the primary, secondary, kernel parameter, and then the parameters that specify the sojourn times in the E and the I class. And this kind of model is very useful for plant epidemics. If we look at something like a disease of citrus, so these pictures that you're seeing there are actually showing citrus cankers spreading through a region of Miami, so you can kind of see the patterns of the streets. The reds are the infectious individuals, then the black is removed, and this is giving snapshots of what the epidemic kind of looked like. And you can kind of see that there is some sort of spatial relationship between the way in which new infections are appearing. And this is a classical example of the kind of situation where we could apply that model. Now, why would be interested in applying a model like that? The kind of eradication strategies that people would use for citrus canker, one common way to approach it would be to use a ring culling strategy. So if you discover a new infection, let's say at some point in your plantation or your orchard, then what you would do is remove all the susceptibles within some radius. And not surprisingly, the radius that you would choose in order to give you adequate control, if you're basing that on the predictions in some model, that is going to be very strongly dependent on the choice of the spatial kernel function. In Miami, we used a 900-foot eradication radius, which actually turned out to be quite controversial. People felt we were removing a lot of citrus trees that perhaps it didn't have to. But clearly, a very important problem is to be able to understand parameters in the spatial kernel function, and also to be able to actually test whether or not the spatial kernel function you're using is appropriate. This is relevant to other situations as well. For example, foot and mouth disease in the UK, Bin Culling's pathogens were used there, and very similar models have been used to actually model foot and mouth disease by people like Chris Jewell and Gareth Roberts. So even though I motivate this using plant epidemics, it's certainly relevant to a whole range of epidemics. So the approach that certainly in our work we've always taken to model fitting is to use the Bayesian approach and really the data augmented MCMC approach. So in a nutshell, this is how this actually works. Let's suppose you lived in an ideal world where you could observe an epidemic and you could see absolutely everything that happens in the epidemic. So X of t is going to be the times and nature of all the transitions that happen up to time t. If you lived in that situation, then you would have a nice tractable likelihood function which would enable you to estimate theta. But in practice, whenever we observe epidemics, we only get partial information on the epidemic. So typical situations might be that you can't see the S to E transitions. You might only observe transitions into the move class, or you might have snapshot data like I showed before. So what you observe is Y, which is some kind of imperfect filtered version of the X of t. So the kind of Bayesian trick is to say, well, let's take all the information in X of t and let's treat that as, if you like, additional unknown parameters. And now what we will look at is the joint posterior distribution of theta and X of t given the observations Y. We can express that as being proportional to the product on the right-hand side. And this term here will typically be tractable, which means that we're able to investigate this joint posterior using, for example, Markov chain Monte Carlo, but also people like Aaron King, Ed Ioannidis have actually used particle filtering. So there's various ways of doing it. One can also take an ABC approach to these problems. But since most of the things I'm going to be looking at are going to be focusing on, if you like, this imputant latent process, then this framework is very useful for the kind of analysis that we're going to do. The kind of Markov chain Monte Carlo algorithms that one can use typically Gibbs sampling, Metropolis, Hastings. We typically have to use the reversible jump methods, which is essentially the Markov chain techniques one can use when the state space of the Markov chain is going to have variable dimension. And that will occur in epidemics because if you can't see the S to E transitions, you will not know precisely how many unseen infection times there are, for example. So therefore, you will have a state space which will have components of varying dimensions. Just a caveat, a very quick caveat, is that even though using these methods, it's possible to cope with the fact that you have partial data, it could be that you have a very nasty posterior distribution and you can have problems with sensitivity to your choice of priors. And this is an example from a paper with Eric Renshaw a long time ago. I was wanting to have a look at a situation where we could apply Markov chain Monte Carlo in order to cope with partial data, but I wanted to have a situation where you had a tractable, analytically tractable posterior distribution. So the simplest epidemic model you might have is the immigration death model, where you start with an empty population and your arrivals would correspond to infection times. That occurs according to a Poisson process. And then there's an exponential waiting time before the removal, which corresponds to the deaths. Now, if you observed all the arrivals and the deaths, you would very easily be able to write down the joint likelihood of alpha, the arrival rate, and mu, the death rate. This is a simulation with alpha equal to 1, mu equal to 0.05. You can very easily write down what that likelihood is, and you would find you got a very nice likelihood of this form here. And it wouldn't really matter what prior you're putting on your alpha and mu. You would typically end up with a posterior that had a lot of mass close to the correct values of alpha and mu. But let's suppose now you don't actually observe the arrivals and all you observe is the deaths. Well, you still have an analytically tractable likelihood for this case. The deaths actually follow a non-homogeneous Poisson process with this rate here, but your likelihood function now becomes very, very badly behaved. You have infinite ridges heading off up here as mu goes to infinity, and there's an infinite ridge heading down here. And that therefore means that actually you'll get some very strange properties in your posterior distributions, particularly if you're using non-informative priors. So just a quick caveat to say that if you're doing this kind of thing, you need to think quite carefully about some of these potential sensitivities when you're using non-informative priors. However, it is possible, and in work with Chris Gilligan's group down at Cambridge, we've looked at this data set and shown that it is possible to capture the dynamics of this using an SEIR spatio-temporal model. Actually, we found that a good fit was using an exponentially decreasing kernel. So our K function looks like e to the minus kappa times distance. Okay, so what are the key questions I want to look at now? Given the importance of spatial kernels in making any kind of prediction, that's a really key part of the model to be able to try and get right. So it's often quite hard to get very good estimates of the parameters there. And a question that people really started to look at probably about 10, 15 years ago was looking to see what would happen if you also had some genetic data on the pathogen. So in other words, as well as observing where infections were, you actually sampled the pathogen from those infections and sequenced the genome. How much improvement could you get if you integrated that into the analysis? So that's one thing I'm going to look at. The other question then I'm also going to look at is how we can try and assess some of the model assumptions that we make and try and refine the model structure. So until COVID-19 came along, this whole question of introducing genetic data into epidemic analysis seemed to be probably the main thing, at least at the conferences that I was going to, that people were talking about. And there is a whole host of literature now on this that takes different approaches, and quite a few packages out there, including things like Outbreaker, for example, Beast, things that can be used to try and integrate genetics. This body of work, some people are coming more from the genetics side and building in the epidemics. We are definitely coming at it from starting from the epidemic model and trying to build in the genetics into the model and seeing if we can integrate the two in some sort of sensible way. So I'm now going to think of the situation where we have some observations which are going to include, let's say, information about which individuals were infectious, which individuals weren't infected. So we might have something like, say, censored infection times. But I'm also going to look at the situation now where we assume that we have some pathogen sequences, and this would be an RNA sequence, which would be appropriate for something like foot and mouth disease virus, which I'll look at later. So we're going to assume that we've got pathogen sequences at some of the sites. Now, if you've got the pathogen sequence, if you look at the phylogenetic tree, that should give you some information on who infected whom. And at the same time, if you have an epidemic which has, let's say, is spreading through some spatial interaction, then the spatial location of infections is also going to give you some information on who infected whom. So the question is, what happens if you try to integrate these two sources of data together? How much better can you do? Okay, so if you're going to extend your epidemic model, you would need to now build in some kind of genetic model. So in particular, since we have a primary infection process, we would have to think about when we have a new infection coming in from the primary infection, what would be the sequence of the pathogen that causes that. So we're going to assume that that's some random perturbation of some master sequence. And then once once an individual has been infected, we now need to think about how the pathogen is evolving within the individual. So I won't go into great details on the genetic model, but basically it's a Kimura model with rates U1 and U2 for the two different kinds of mutations that can occur, the transitions and transversions. So it's essentially saying that any site on the genome, mutations are occurring according to a Poisson process for the two different kinds of mutations. We need to make some further assumptions, though, to get things to be analytically tractable for our likelihoods. And the assumption that we make, in common with a lot of other workers in this area, is the idea of a single dominant strain. So in other words, we assume that within a host, there's only a single strain of the pathogen which is dominant at any given time. And effectively, in this model, we're ignoring strain diversity within hosts. And then a final assumption that needs to be made explicit is that we're assuming that the evolution of the pathogen is not happening sufficiently fast that it affects the transmissibility of the pathogen. So transmissibility is not going to be affected by genetics. So if we're going to extend this whole data augmented MCMC approach to try and tackle the problem where now we're building in the genetic data, how do we do that? So my data Y now comprises the epidemic observations plus some of the observed sequence data. So now, I'm now going to have to look at a rather more complicated data augmentation process. I'm going to include the unobserved exposure times. I'm going to include the unobserved times when infectiousness occurs. But in addition, I'm also going to include the transmission graph. So psi is going to denote the graph which tells me who infected whom in the epidemic. I'm also going to include the transmitted sequences. So as you can see, I'm augmenting my observations with a lot of additional material. So the question is this actually feasible to do? And now, in terms of the parameters, I have my original spatio-temporal XEIR model parameters, but now I have my genetic parameters for the transition, transversion rates, the master sequence, and then this probability, which is going to control the perturbations of the master sequence that form the primary infection. So the question is it possible to develop samples that can explore that effectively? So you can read the papers on this to go into details on the algorithm, but just a very quick indication as to what the main challenges is. The problem is that if you propose a change to the source of an infection, then you need to make sure that you're also proposing a change to the transmitted sequence. So this picture here tries to summarize what's going on. Let's suppose you've got a very small population, just 4 individuals. Think of time as going in this vertical direction here, and each individual becomes infected during this epidemic. And the blue lines indicate the period of time where the pathogen is, if you like, in the individual and evolving. So this would be, if you like, the union of the exposed period and also the infectious period. And let's suppose in this situation, what we're doing as we're running the MCMC is we're imputing the source of infection, we're imputing the infection times, and we're also going to be imputing over here the transmitted sequences. Now, let's suppose in this particular configuration, I'm currently assuming that X3 was infected by X2, but let's suppose now I propose a change to that source. Then at the same time, I have to propose a change to the transmitted sequence if I'm going to have any chance of accepting that. So the key thing in the algorithm was a way of actually trying to come up with a method of jointly proposing changes to the source of infection and the transmitted strain that would give you acceptable or reasonable acceptance probabilities. So I won't go into the detail on that, but happy to answer questions on that. So it is possible to build this into a data augmented framework and actually apply this to real world data sets, but actually to test it out initially, we looked at simulating some epidemics and then trying to see how well we could actually recover the parameters used in the simulation, and in particular trying to look to see what would happen or how much better we could do in terms of characterizing the spatial kernel if we included the genetic information. So the simulation is chosen to reflect the foot and mouth disease situation. So we've got 150 sites which are just uniformly distributed over a square region. So think of those as being 150 farms, if you like. Then we simulate an XEIR spatial-temporal model with an exponential kernel. And we're going to assume, as in the case with foot and mouth data set we were working with, that you have a sequence of length 8,000 for the genome. And we assume, again, that the primary infection sequences are drawn as random perturbations of some consensus sequence. So in this simulation study, we looked at 2 cases where there was different primary infection rates. Now, the higher the primary infection rate, this means the more, if you like, kind of independent introductions you're going to get. So you're going to get more independent clusters appearing in your epidemic. And in the 2 different primary rates, we used that to generate an epidemic with 3 separate clusters, so 3 primary infections, then with everything else from secondary infection, and also one with 6 clusters. And they were run until nearly all individuals were infected. And we assumed that you could see that the STE transitions are unobserved, and that we had some sensitive information on the E to I and the I to R. So what we're interested in is, first of all, how well can we recover the transmission graph when we throw in the genetic information, and the way in which we can characterize that is by looking at what we call overall coverage rate. So overall coverage rate is just the proportion of individuals in the imputed transmission graph where the source is correctly identified, and also the cluster identification rate. So this is really a measure of how well an individual infection is associated with the correct primary infection. But another question is how much is the precision of your parameter estimates affected by the amount of genetic sampling? So we looked at the situation where basically you sequence all of the infections, so 100%, 80%, 50, or 0%. So 0% would just correspond to the analysis without any genetic data at all. And what we can see is actually when it comes to looking at the coverage rate, so how well you identify the sources of the infections. So the way in which we generate these is as we're running the MCMC, we take the graph that we've generated, we look to see what the coverage rate for that graph is. And then as we're running the MCMC, we generate then the stereo distribution of that coverage rate. And you can see that with 100% sampling, you get actually very good coverage rate, and it deteriorates obviously as the sampling goes down. So with less genetic data, you get poorer performance, and that's the same for both the 3 and the 6 6 cluster epidemic. We were interested though in how well your estimates of the spatial kernel parameter, how they improved with the genetic data. And in particular, what you can see is that with no genetic data, you're not very certain at all about the spatial parameter, whereas as you include the genetic data, you now start to get much more precise estimates of what that spatial parameter actually is. So that's good. The dotted lines show you the values that are actually used for the simulation. So that was quite promising. Cluster recovery rate, to save time, I won't go into details on this, but simply to say that we see the same pattern that for the 6 cluster epidemic, we find that if you include all the genetic data, so samples from every host, then you actually recover the 6 clusters quite nicely, and that each infection is being correctly associated with the correct primary infection, although without any genetic information, you'll find that it's hard there's some clusters it was very hard to correctly identify the primary infection. So basically the size of the dot, each dot corresponds to an infection, and the size of the dot is telling you the posterior probability that it was correctly associated with the correct primary infection. So to save time, I won't go into that in a lot of detail. We also applied the methods to a foot and mouth data set, which I'll say a little bit more about later, time permitting. And they were also picked up by Simon Firestone in Melbourne, actually, who's now built this algorithm into a package called Boris. And he did a comparison of a lot of the different approaches to combining genetic and epidemic data. The method I've described turned out to be good at reconstructing transmission trees, not quite so good at estimating the molecular clock rates. Other more sophisticated genetic methods are better, but that's perhaps not surprising given that we were starting from the epidemic and epidemic model and then adding in the genetics. So I think we had to make a lot of simplifications there. So natural question to ask is, how could you refine that model, and what's the big weak spot in the model? Well, I would say that this single dominant strain assumption, which essentially ignores any pathogen diversity, is a major simplification that's unlikely to hold in practice. So a question would be, is it actually possible to detect evidence for that mis-specification without adding excessive complexity into the algorithm? So how can we do that without having to for example, look at a much more complex model? Is there a way we can do that just as a simple add-on to the existing analysis that we're doing? So that's what I want to talk about in the next part of the talk, is to look to see how we can use essentially methods that are rooted in posterior predictive checking in order to try and test whether these assumptions actually hold. So I'm going to say a little bit now about the approach that we take. There's different ways in which you can think of this, non-centered parameterizations, latent residuals, functional models. I tend to think of it as in the form of a functional model, a very simple idea. Anybody who's, when I first started doing computing, a lot of the computer languages only ever had a uniform 0-1 random number generator, which meant that any stochastic process you simulated, essentially you had to do that by transforming samples from a uniform 0-1. So that's essentially the idea in a functional model representation. So in a functional model representation, we assume that we have some stochastic process, which let's take that to be IID uniform 0-1, and then we have our model parameter, and then we express the outcome of the process that we're interested in modeling as some deterministic function of R and theta. So think of, in the epidemic context, X is all my epidemic data, and I'm thinking of X then as just being a function of R and theta. So a very, very simple idea. And how can this then help you when you actually want to come and test the model? Well, if instead of looking at pi of X, pi of theta and X given Y, which I do in my my data augmented MCMC, if I was actually then to impute from the theta and X, if I was to impute what the R was, then I could investigate pi of theta and R given Y, and the imputed R, I can treat that as some kind of latent residual process, which if my model is correct, the distribution of those latent residuals should conform to F of R. So one way in which I can try and test the model is to impute this process here and test to see whether or not it matches the process defined by F. And in these examples, it's always going to be uniform 0-1. So one way in which you can think of this in terms of posterior predictive checking, I find it useful to think of it in this kind of two observer process. So you're the Bayesian B and you've fitted this, or you're going to fit this model, which has a lot of assumptions in it. And your kind of classical conscience says, well, I think there might be something wrong with the model. And so think of C as being conscious. And it says, once you get Y, what I want you to do is impute the results of a classical test of fit of your imputed R to this distribution F of R and work out a p-value. And then if you look at the posterior distribution of that p-value, then that posterior distribution gives you a measure of how much these beliefs conflict with the analysis which has been done by C. So a typical or the usual posterior predictive p-value would simply be the posterior expectation of P of R, but actually it's sometimes quite useful to think of the full posterior distribution of P of R, because that can give you some very useful information, which hopefully I'll get time to show you towards the end of the talk. So that's the general idea. So in particular, you might be interested in knowing, okay, what's the posterior probability that this p-value is less than 0.05, and if that's large, then that is telling you that with a high posterior probability, this classical observer has kind of rejected the model for R, and that calls your model into question. So the advantages of this is that you can design this setup in any way you want, so long as you preserve the sampling properties of Y given theta. And in particular, you can tailor that R to suspected mis-specifications. You're also able to use tests then by applying the tests to the latent R. They can be analytically tractable. But one of the problems that you run into is that any latent quantity that you impute is going to be imputed using the model that you've fitted. So therefore, it's going to reinforce the model. And that means actually you might get a better test by using less imputed information than more. So how would we do latent residuals for the SDS model? So I'll speed up a little bit a little bit here. But we now have this model that has the epidemic part of it, and that also has the genetic part. So how would we come up with a functional model representation? Well, in my functional model representation, I'm going to have basically 4 independent IID uniform 0-1 processes, and I'm also going to have 2 independent IID uniform 0-1 processes. And the R's are going to be the one that control the epidemic events. So once I've generated, if you like, the epidemic using the R's, then this effectively gives me a kind of lattice on which the genetic process is going to develop, and then I'm able to if you like, work out the realization of the genetic process once I've got the epidemic process. So just very quickly, in this process here, the R1s effectively control the waiting time for each new exposure to occur. R2s, which I may come back to later, actually they're going to control the particular infection susceptible link that's responsible for each exposure. R3 and R4 are just going to give me respectively the quantiles for the sojourn time in the E and the I class. And then for the genetic side of it, how does the S1 and the S2 actually come into it? I'm just going to skip over this. So the S1 and the S2 are going to be, they're going to control the mutations that occur within this lattice. So let me look at this particular individual here. So this was infected by this individual, which transmitted that strain. And now what I need to do is come up with the mutations that occur during this period here. This is going to be the period where I actually observe the strain in this infected individuals. So S1 is effectively just going to determine the quantile for the number of mutations that occur. And so that's just the quantile in some binomial distribution, and S2 would be responsible for actually determining which sites actually are actually mutated. But actually, in the analysis that we do, we just worry about the S1, so we just focus our attention on this S1 residual. So within the MCMC analysis, it's fairly straightforward to actually impute the S1s when you're doing the MCMC. So that means that we can effectively impute the S1 variable for each of the periods where we have to, if you like, model the evolution of the pathogen. And that's how I would then generate a set of S1s, which I'm actually able to test using, in our case, an Anderson-Darling test. So I will go a little bit quicker because I realize that time is running short. So, but we can do something a little bit more powerful than that, because we're actually at liberty to focus in on certain residuals in the process here. And the natural question to ask is that in what circumstances would you be most likely to observe deviation from this single dominant strain model if you actually did have within host diversity? So this diagram attempts to capture what's going on, but I'll just jump straight to look at case 2 here. Let's suppose we have an individual which got infected with this light blue strain at time t0, and then let's say at time ta in the future, it infects another individual, so it passes on this green strain, and then at time td, let's say it's sampled, so the red strain is what we actually, the red sequence would be what we actually observe. Now, if you have within host diversity, so if there's actually a population of strains within the individual at this point, then that could be manifest by having actually quite a big difference between the two sequences, the red one and the green one. So when we have these two events which are close together, then that's when we might expect to see this within host diversity really being manifest. So what we can do is, before we even think about the strains, with the epidemic part of the model, that specifies the timings of all the events. So we could actually assign a mark to each link, where the mark would be high when I had a situation like this, where we had two events that were occurring close together in time, some considerable time after the initial infection. So I can assign a mark to each link of this form, and then I'm quite entitled to base my test on a subset of those residuals which correspond to the links with the highest marks, because that's where I might expect to see this deviation from the uniform 0-1. So just very quickly, we tested this out. using a similar model to the one from the 2015 paper. So this is that paper from last year with Max Lau. Again, we simulated epidemics similar to that 3-cluster setting, but this time we simulate within host molecular evolution 2 different ways, with the SDS model, and then we also did it with a birth-death mutation model that explicitly would give us a population of strains. So this is a model now that actually has within host diversity. What we then do is fit the SDS model, impute the residuals, and then look at the stereo distribution of the p-values that one actually gets. And we assume observations similar to what we did in the 2015 paper. We assume we don't see the S to E transitions, but we can more or less see the E to I and the I to R. So just very quickly, I'm focusing in here on the situation where we fit the SDS model to the case where we have within strain diversity. And this is showing the cumulative distribution function for the p-value that comes from the Anderson-Darling test applied to the genetic residuals. So what does that, I realize that time is now getting short, so I'm going to probably jump towards the end of the talk quite quickly. But what we find is that, yes, we are able to find strong evidence. So when we use only those residuals corresponding to non-zero marks, we actually get more evidence against the model than we do than when we use all the residuals. But in both cases, there's substantial amounts of evidence that the p-value should be less than 0.05. So we are actually then able to pick up some evidence. But it's interesting that using the non-zero marks, so using fewer residuals systematically, is giving us more evidence here. We apply that also to the foot and mouth disease data set, where this was a small cluster of infections from the 2001 epidemic, much smaller, only 15 infected farms. Typically, that means that we only had about 30 residuals. And in that case, it turns out that using all of the residuals gives you more evidence than looking at the residuals corresponding to the non-zero marks. So more is more in this case. But I should stress that it would be inappropriate to work out what's the optimal size of residual set to take and choose the one that gives the most evidence. You would really have to specify that a priori. So very quickly, I'm going to jump towards the end of the talk now, actually, because I realise that I'm kind of running out of time. We did look at a method of trying to build in strain diversity in the model that we fit using a serial likelihood approach, which you can read about in the 2019 paper. But just in the very last couple of minutes, I want to go back to the likelihood or the problem of actually testing the spatial kernel and just make a couple of comments on the work with David Thong. This is work that's, we're just revising the paper at the moment. So this is looking at a different approach to trying to test the model, where we actually explicitly impute a likelihood ratio test. So in what I was doing before, I was looking at classical tests which were applied to imputed residuals, but there might be more powerful ways of doing it, where we could actually specify an alternative model and try and look at some latent likelihood ratio tests. So in this approach, and we've specifically applied it to testing for spatial kernels, I'm now going to assume that the Bayesian B has the model that they're fitting, but there is an alternative model which is specified. And in this example, it's going to be a model where it's identical to this model here, except that it has a different spatial kernel function in it. So it's an SEIR, spatio-temporal model, but they only differ in terms of the spatial kernel function. So again, the Bayesian B can investigate this joint distribution of theta and x, where x is all the latent events that aren't observed using MCMC. And then for each theta I and xi, what they could do would be to impute the value of a generalized likelihood ratio statistic, where they take pi naught of xi given theta I, and then divide by pi 1 of xi, and take the maximum likelihood estimate for theta 1 in the alternative model. So effectively, they're imputing the value of a likelihood ratio statistic, which somebody who observed the full epidemic trajectory would calculate. And then you can look to see what this p-value looks like by investigating what this, the probability that you got a smaller value than the one observed over repeated sampling. So just very quickly, we've compared that technique for trying to detect the specification of a spatial kernel with the technique that we looked at with Max Lau in 2014, infection-linked residuals. I haven't really had time to actually describe those, but I'll just kind of cut to the chase in the simulation study where we have a population of size 1,000 and we simulate our epidemics using an exponential kernel. So this kind of represents the citrus canker setting. We're going to assume that we don't observe S to E, but we do observe the other transitions. What happens if we fit the wrong model? Well, if we fit a model where we have a power law of decay, that's a severe misspecification. If I fit a model of this form, that's going to be a relatively mild misspecification. Are we able to detect these misspecifications using the latent likelihood ratio test? And what we find is that when we have the milder misspecification, just summarizing those, I'll be happy to send anybody that's interested a copy of the paper. But what we actually find is that compared to the residual-based methods which we've used in the past, the likelihood ratio method actually gives you, is able to find more evidence against the misspecified model. So we're reasonably hopeful that actually these kind of imputed likelihood ratio tests give you a much more powerful way than the residual tests when you're trying to detect misspecification, which is a relatively mild misspecification. So remember, the true model is an exponential e to the minus kappa d. Here we're fitting something that looks more like a Gaussian, but it's still exponentially bounded, where having a power law decay is very much a severe misspecification. So we feel the results there are actually quite promising. I realize I'm out of time, so nevertheless, in this setting, we can still get some information about how the model's misspecified from the residuals, but I realize I've actually probably gone on longer than I should have done, so I will more or less stop there. Conclusions, I think using latent discrepancy variables is very useful in terms of testing models, because it means that essentially you can do it as a fairly simple add-on to your Bayesian analysis, but there's still a lot of very challenging problems in this whole area of genetics and epidemiology. So anybody with an interest in MCMC sampling, there's some very interesting problems there, sort of non-standard problems, which you can get your teeth into. So I'll stop there, thank you.
Thank you so much, Dr. Gibson, for the wonderful presentation. So we have a question here. So Dr. Dearden, can you just unmute yourself to ask the question?
I can. Thanks, Gavin. That was a really nice talk. I wondered, with the joint epi-phylogenetic models, what sort of computational power do you need to fit these? Are you running these on a cluster? And
yeah, in the 2015 paper, I'm trying to remember the figures. It was about sort of 18 hours on a cluster. So it was a lot of computation. I mentioned that algorithms now in a package called Boris, which Simon Firestone put together. I'm not sure what the running times of that would actually be.
Okay.
But one thing I would say is that in David Thong's thesis, a lot of the methods that he used, he then implemented on GPUs, and that was really, I was amazed that that reduced sometimes by a factor of 200. So yeah, the run times are long, but I think if you can parallelize the likelihood calculations, then that can really bring it down radically, because you can then exploit the GPUs.
Yeah, I sometimes feel like I have a hunch as well that maybe sequential Monte Carlo would do better when you have these really horrible correlations. I think
you might be right. I mean, I guess I've kind of often stuck to the data augmented MCMC. I mean, all I would say is that so long as you're still able to impute the latent process, which is kind of central to these techniques, then yes, it may well do better. But I would put my hand up and say that yeah, we haven't maybe optimized in terms of the running times, but I would say that they're certainly practicable for for these kind of applications.
Okay, yeah. I mean, I'm an MCMC guy as well, but okay.
Okay, great. Yeah, thank you so much again, Dr. Gibson.